The role of glycoproteins in adhesion by Gerardo Garcia. The extracellular matrix is held in place by adhesive glycoproteins. Adhesive glycoproteins such as fibronectin and laminin anchor cells to the extracellular matrix. Fibronectin links cells to the extracellular matrix, while laminin attaches cells to the basal lamina. Cells use cell surface receptor glycoproteins called integrins to connect to the extracellular matrix. The three main players responsible for adhesion are fibronectin, laminin, and integrin receptors. The fibronectin structure consists of two nearly identical polypeptide chains joined together by two disulfide bonds near their carboxyl ends. Each polypeptide is folded into a series of domains linked by short flexible segments. The domains have binding sites for extracellular matrix components or for specific receptors on the cell surface. The laminin structure consists of three long polypeptides denoted alpha, beta, and gamma. The polypeptide chains are held together by disulfide bonds. Domains on the ends of the alpha chain are recognized by cell surface receptors. Those at the ends of the two arms of the cross are specific for type 4 collagen. The integrin structure consists of alpha and beta subunit transmembrane polypeptides that associate with each other non-covalently to form a binding site for the ligand on the outer membrane surface and a binding site for linker proteins on the inner membrane surface. This fibronectin receptor, alpha 5 beta 1, has a binding site for fibronectin on the outer surface and a binding site for talin on the cytosolic side of the membrane. Fibronectin is a bridging molecule that attaches cells to the extracellular matrix. It is found in cell surfaces, extracellular matrices, and in blood. It promotes blood clotting due to having several binding domains that recognize fibrin, the blood clotting protein, and it can attach to blood platelets to fibrin as the blood clot forms. Laminin is present in the basal lamina and binds cells to it. The basal lamina underlies epithelial cells and separates them from connective tissue. Basal lamina surrounds muscle cells, fat cells, and Schwann cells and serves as a structural support that maintains tissue organization and is a permeability barrier that regulates the movement of molecules as well as cells. Integrin receptors are receptors that bind to fibronectin or laminin molecules. They integrate the cytoskeleton with the extracellular matrix is located on the surface of cells. Weak adhesion can cause severe conditions such as junctional epidermolysis bullosa, aka JEB. It is caused by a mutation in one of the subunits of a particular type of laminin. The basal lamina associated with the attachment between the outer layers of epithelial cells and underlying tissue failed to form properly. Patients experience severe blisters over a large portion of their bodies from any type of friction against their skin. This disease cannot be diagnosed in utero, only immediately after birth, since blisters may form on the baby. Patients usually die as children, and only systematic steroids can produce relief. So the picture on the right is the hand of a 3-year-old girl that suffers from junctional epidermolysis bullosa. The study that I chose was the overexpression of laminin in breast cancer. Laminin expression in normal breast tissue and in breast invasive ductal carcinoma were observed. Immunohistochemical method was used to detect levels of laminin. Squares A and B represent normal breast tissue, while C and D represent breast cancer tissue. The diagnosis of the breast tissues was confirmed by two senior pathologists with hematoxylin and eosin staining. The colorless color is negative, pale brown positive, and brownish black strong positive for levels of laminin. C and D showed that laminin was strong positive in invasive ductal carcinoma. So what this study tells us is that laminin is overly expressed in breast cancer, and it is involved in breast cancer invasion and metastasis. An overexpression of laminin can be used to determine whether a tumor is malignant or not. The basement membrane is recognized as a cancer cell defense barrier, which inhibits the local infiltration of cancer cells by inhibiting the adhesion of cancer cells to it. Cancer cells break through the basement barrier, which is composed of basal lamina and lamina lucida. This causes the tumor to develop. Overexpression of laminin make it easier for cancer cells to migrate and degrade the basement membrane. So based on this study, I have a question. What kind of treatments will help to inhibit the overexpression of laminin in breast tissue?